We are going to talk about the microbiome and some of the roles that it has in cardiovascular disease. And I always tell people this when I start out. To be clear, I'm not a microbiologist, okay? But I think that's actually a good thing. To me, I use microbial analysis as a tool to explain other things. When I get calls all the time, when people see on the website, they're like, hey, can you sequence this and do all that? And I'm like, no, I actually don't know how to do that. I don't want to know how to do that. Do all that through collaborations. But we do use those data and that information. So I want to talk today about something called Trimethylamine N-oxide, TMAO. Has anybody here actually heard of this before? Colin, okay, a couple of you, right? So this is kind of one of those hot topics, and it's very interesting. It actually, if you look at a Venn diagram, it's actually right at the intersection of nutrition, the gut microbiome, host metabolism, and I actually should have put another one, I should have said health here, okay? So this slide is copied directly from my interview presentation like a year and a half ago. Oh man, that's been a long time. So just a quick overview of the gut microbiome. This is what I tell people on an airplane when I sit next to them and I tell them a little bit about what I do. They always say when you're writing an NIH proposal or any kind of proposal, you want to have that lead-in statement that's just that attention getter, okay? So what I tell people is if you think of the bacteria that live in your gut as an organ, okay? It's an organ that is not made up of mammalian or human cells, okay? But it does have the same potential as an organ. It has metabolic functions, endocrine functions, immune functions, barrier functions, and things like that. And some interesting statistics. So I think any of you that have kind of read anything about this field know that there's over 1,000 species of gut microbiota that they've identified, okay? And this is not your traditional microbiology where you culture it on a plate and you do streak plates and try to get little colonies, and then you take those colonies and you'll do like, are they acid fast, are they acid producing, what kind of properties? You actually can't grow most of these bacteria outside of the gut. So we rely strictly on molecular techniques to look at, these are all just based on molecular biology, sequencing, and looking at their genes. Okay? These are not things that you can isolate in the lab. Generally speaking, you can get some of them, okay? But the advances in molecular biology techniques and bioinformatics in the last 25 years have made it possible to study this, okay? So some interesting facts. There are about over 3 million non-redundant genes in the gut microbiome genome. And interestingly, that's 150 times the number of genes in humans. Okay? So your gut microbiome, and I'm not even talking about skin or other surfaces, I'm just talking about the gut, has 150 times more genes than you do. So if you're talking about therapeutic targets and things like that, the gut microbiome is very interesting because there's just more stuff to target. Okay? Prokaryotic cells, right, these cells that don't have nuclei, they actually outnumber us 10 to 1. Okay, and that's because these cells are a lot smaller than mammalian cells. They're a lot simpler, okay? And so another statistic that I tell people is that, and this is what really grosses them out, and this is if I want them to stop talking to me on an airplane. I say, well, if you defecated in a jar and dried it in your oven, and they're like, what? I say, if you dried that, and then you weigh, and if you could figure out, if you could separate the bacteria from the other stuff, that fecal matter would be 40 to 60% dead bacterial cells. Okay? So the message I'm trying to get here is there's a lot of it, okay? And it's important, okay? It's a great target, it's an attractive target for phytochemicals or other dietary strategies because there's just a lot of cells, a lot of pathways, and for us, for those of us that work in the phytochemicals world, one of the things that we worry about is actually delivering those compounds to the tissue that we want to have an effect. Well, for things that are in these cells that just live in the GI tract, there is no barrier, right? Other than the actual cell membrane. Your gut microbiome is bathing in the things that you eat. More so, in fact, than our own cells, okay? The things that we eat come into contact with the gut microbiome at higher concentrations than they do with any cell in our body except for our gut epithelium. Okay? So it's a really attractive target to deliver phytochemicals without having to worry about things like barriers to absorption or phase 2 metabolism and efflux and things like that. Okay? So that's why we're interested in the gut microbiome. Now, anybody that's been doing any reading or going to conferences knows that in the last 15 to 20 years, this has been what I think is probably the hottest area in nutrition and phytochemicals. The problem is that anytime there's a cool, new, shiny object, people want to use it. Okay? So we know that the microbiome metabolizes phytochemicals and that phytochemicals also modify microbiome composition. And there have just been these massive numbers of papers that talk about how some phytochemical alters the composition of the gut microbiome. And they use 16S, the ribosomal DNA, or 16S ribosomal RNA gene, okay? And they just tell you, oh, there's differences in abundances of different bacteria. And there are tens of thousands of these papers out there. Most of the time, that's very descriptive in that we don't know what that actually means other than it changed the composition of the gut microbiome. Okay? So where the field is heading is we now want to know what are the functional implications of those descriptive changes that we've seen. So the question I always ask people is, so what? There's a different composition of the gut microbiome. Who cares? Okay? It doesn't matter if we don't know what those cells are actually doing. Okay? Right? If I have a basketball team and the center gets injured, I put in another center. It has a different name, but it's still doing the same thing that a center does, right? It's standing in the middle, rebounding, right? So the question is, what we want to know is, just because it's a different name of a bacteria, what is its metabolic potential? What is it doing? Okay? And that's generally where the field is heading, I hope. Okay? So getting away, and there's nothing wrong with this descriptive work. That was what had to be done first. The problem is we need to move past that and actually start exploring deeper and deeper. But sequencing has become moderately more inexpensive, and so it's being used more and more. And the problem is there's just not a lot of meaning behind it sometimes. Okay? So just to give you that perspective, I will say that our knowledge of microbial community structures, it's not going to become less important. We just need to understand it in the context of function. Okay? So I wanted to talk briefly, and I really wish this were my paper because it was in Nature. You know, we all want to get that Nature paper. So in 2011, and it's interesting to me that this is so recent. I had actually got my first faculty position in 2011, but this paper came out that has just wildly transformed the field of diet and cardiovascular disease, specifically related to nutrients in the microbiome. I guess you can't really see this here, but Stanley Hazen is this PI at the Cleveland Clinic. And what they did is they took a metabolomics approach to understand what metabolites were predictive or diagnostic or related to cardiovascular disease. Okay? And I really encourage you to go read this paper. I can send it to you. But basically what they did is they had some cohorts, and they used some metabolomics techniques and to correlate these metabolites with risk of cardiovascular disease. And what they were able to do is they were able to identify these 18 analytes. Okay? And what you see here is they've got this really interesting figure where they have the mass to charge kind of spectrum of these analytes along with, on the x-axis, and they have the odds ratio. In other words, if you compare the highest quartile to the lowest quartile, what is the relative odds that people with the highest quartile have higher cardiovascular disease risk than those on the lower? Okay? Usually when you're looking at epidemiology, you talk about, maybe a 5% increase or 10% decrease. Okay? We're talking 10 to 30-fold greater risk associated with these 18 metabolites. Okay? And then they looked and said, how are these metabolites related to each other? We want to focus on a pathway. And these dark circles, okay, at these masses, these metabolites were highly correlated with each other. The other ones appear to not be related. So they said, okay, we want to focus and figure out what the heck those things are. Okay? Oop. Sorry, I wasn't doing my animations. Okay? So one of the things that they focused on, there's this metabolite at like 104. There's 76, 104, and I can't remember what this other one is. I think maybe 1 something. Okay? But choline was 104. And they figured out that if they administered choline by oral administration, they also got this 60 and this 76, which turned out to be trimethylamine and TMAO. Okay? However, what they realized is that if they gave it IP, they don't get either of these. Okay? So the first thing they said was, okay, that means that these metabolites are related to the gut, probably the gut microbiome. We don't see them unless we administer them orally. Okay? All right, so then the next thing they did was they wanted to test that hypothesis. So what they did is they did the same experiment where they administered choline orally, and you see TMAO being formed here. Okay? Then what they did is they administered antibiotics to suppress the microbiome, and you see TMAO formation essentially goes away. Then they allowed the animals to recover their gut microbiome, and you see that TMAO production after oral gavage of choline is restored. Okay? So through these two experiments, they were able to show these metabolites that are associated with this higher risk of cardiovascular disease, they are dependent on the microbiome. Okay? You can only get them if you administer them orally, and antibiotics take them away. Okay? So the next thing they did is they looked at these metabolites, and they just looked at the odds ratio. If your concentration, and this is in a cohort of about 1,800 humans, what is the relationship between increasing concentration of these compounds and the odds ratio of incident cardiovascular disease? And you can see choline and TMAO, I mean, these are just an epidemiologist's dream, right? They're so tightly correlated. Okay, these are 95% confidence intervals. And basically what that means is as your circulating concentration of choline and TMAO goes up, your risk of cardiovascular disease goes up. Okay? So they're starting to put these pieces together. Okay? Those metabolites are associated with cardiovascular disease. So then they did an animal experiment in mice. The ones I'd like you to focus on was these are just mice fed just a normal chow diet. Then when they supplemented them with choline, you could see the overall area of aortic lesions goes up significantly. And then they said, well, can we get the same thing by just giving them TMAO? Even though we know it's formed in the diet, what if we just give them TMAO and you see the exact same thing happening? So they've then shown in mice that these compounds that are of microbial origin, well, TMAO is, right, that are associated with cardiovascular disease risk in humans, if we give them to mice, we get greater atherosclerosis. Okay? All right, so then they said, well, what happens if we plot out how does TMAO concentration correlate to atherosclerotic lesion area? Nice, tight, strong correlation. Okay? In humans, I keep forgetting to do my animations here. I apologize. Okay? In humans, they showed basically the same thing, that the circulating concentrations of TMAO were related to the number of locations of coronary vessel disease that you had. So the higher your concentration of TMAO, the more likely you were to have multiple locations of severe vascular disease. Okay? The last thing, they also showed that this was related to a liver enzyme called flavin monooxygenase 3. They showed in these mice that lesion area was strongly correlated to FMO3 expression, and TMAO concentration was also correlated to FMO3 expression. Okay? And they also, this is not to get into this, but this was basically showing that this locus where this gene is located, the genotype of this locus is also strongly related to lesions. Okay, so they've showed then here that this liver enzyme FMO3 is also related to microbial-associated TMA production and subsequent atherosclerosis. Okay, I'm sorry, I need to remember to do my animations here. And then finally, they said, okay, what happens if we give animals choline and or antibiotics? Okay, they showed that when they give animals choline, if you compare the open bars here, significantly increase both free cholesterol or total cholesterol and foam cells. And then when you add antibiotics, those negative effects go away. Okay? So why did I show you this? Number one, this was such a landmark paper in the field, okay, that I think it's important for you to understand because these same experiments are going to be used in subsequent papers. Also for the students and others that are here, and even for myself, I think it's so instructive to look at these just really elegant experiments and how they're presented. That's just kind of what you should strive for with your papers, is to keep your figures simple, easy to interpret. Okay, you see they don't have 56 different colors and all these other things, and they're very, very simple and easy to interpret. And you can see the logic in their experiments that they presented. Okay, so the summary of this then is that we now know, well, we've known about this pathway for a while, but we now know that this microbial pathway whereby substrates like betaine, phosphatidylcholine, choline, and carnitine are metabolized and they release this trimethylamine. It's simply a nitrogen with 3 methyl groups attached to it. That's the microbial pathway. This trimethylamine is then absorbed, and in the liver, flavin monooxygenase 3 adds this oxygen, creates a positive charge here, and this molecule, trimethylamine N-oxide, okay, is then when you consume things that are rich in betaine, choline, phosphatidylcholine, and carnitine, so meats, fish, eggs, dairy products, you get elevated levels of these metabolites, okay? So again, this is not a new thing, okay, and I just couldn't resist, right? You guys have seen the movie Poltergeist? I had to do this. Okay, so the point I want to make with this is these are not, we've known about these compounds for a while, okay? What we didn't know is their role in cardiovascular disease. So people that have been studying fish and seafood have known about trimethylamine for a long time, TMAO for a long time. That's a younger version of Dr. Neilson, circa 2010, okay, the pre-weight loss, okay? But marine organisms, particularly fish that live in deep water, they actually make TMAO naturally, and what it does is it counteracts the effect of salt and pressure. If they didn't have TMAO and other things, you would have these shriveled up, crushed, like dehydrated fish at the bottom of the ocean, right? Because the salt in the water would just pull all the water out of their systems, okay? It's actually responsible for off odors and spoiled fish, but the opposite happens. And I told my wife the other day, I said, I'm looking for the grossest picture of rotting fish that I can find. And she's like, why would you want to do that? I'm like, this is my brand, okay? It is who I am. So in these fish, you actually get the reverse happening. So we talked about how the gut microbiome can release TMA, which then gets made into TMAO. Well, the fish have TMAO, and there are bacteria that can degrade that back to TMA, okay? So what they do is they go backwards and produce TMA, okay? Does anybody have any tri or diethylamines in the lab? Does anybody know what they smell like? They smell like rotting fish, okay? So pro tip, go to Publix, get some fish at the counter. If you want to buy fish, say, hey, can I smell it? If it has a smell, don't, right? It shouldn't have any, it should basically be odorless. So we've known about this for fish. Has anybody heard of fish odor syndrome? Trimethylaminuria. Has anybody heard of this? This is a pretty rare genetic disease. They actually don't know how often it occurs. But in these individuals that have this, they are actually not able to convert trimethylamine to TMAO because they have mutations in their FMO3 gene, right? And so instead of producing TMAO, which is rapidly excreted, they accumulate trimethylamine and therefore they smell like rotting fish. It's actually quite a terrible disease to have, okay? You have to use things like acid soaps and be really careful not to sweat and things like this. So we've known about this for a while. The TMAO and TMA are not new. What is new is their role in cardiovascular disease. Okay? So we know now, based on a bunch of research that I'm not going to present, that TMAO has deleterious effects in terms of creating foam cells, altering the macrophage phenotype, endothelial cell inflammation, enhanced adhesion, platelet activation and clotting, and that basically TMAO can contribute to the development of atherosclerosis, heart failure, kidney disease, thrombosis, and actually now I'm part of a group that's actually, and this is not something that we came up with, but we're working on, there are signs that it can actually disrupt glucose metabolism, okay? So it's just crazy to think though, and again, some meta-analyses have then confirmed that circulating TMAO is in fact related to risk of all-cause mortality and incidence of major adverse cardiovascular and cerebrovascular events, okay? And actually it appears that intake of these compounds themselves, carnitine, choline, and so forth, it may not be the primary problem. It's an altered ability to convert them to TMA and TMAO that's actually the problem, okay? I'll talk a little bit more about that later. Okay, so just to visualize this, right? This Hazen paper came out in Nature in 2011, and it's actually easy. If you go to PubMed, you can export a CSV file of publications by year if you ever want to make a graph that looks like this and then just put it into Prism or Excel or whatever. So there are these TMAO papers that were out there. They're mostly about fish, marine organisms, things like that. If you look at TMAO and cardiovascular disease, there was like, this bar is a 0, that's a 1, 1, 0, right? And then that Hazen paper came out, and you don't usually see a curve like this for a topic in the literature. Just to give you an idea, if you go to these phytochemical conferences, like T and cardiovascular disease, right? Colin, Marianne, you guys see these. Every conference you go to, right? If you look at that, that's a fairly developed field. The number of papers per year isn't really changing. But this field, just in the last 8 years, has actually just exploded, okay? And that's one of the reasons myself and some of my colleagues have taken an interest in this. It's really an opportunity to be on the cutting edge. All right, we're not obviously publishing the Nature papers, maybe someday, right? Hopefully right before I go up for full professor, right? I'm not going to count on that one, okay? But just to give you an idea of kind of how hot this field is, there's no sign of a downturn yet in terms of how many publications are being produced per year in this area. Okay, so I got into this, and this is initially my role in this area. So there's this guy named Kevin Davey at Virginia Tech who's a cardiovascular physiologist, and one day in 2015 he sent me an email, and Colin knows this, and Marianne knows this. Hey, dude, can you guys measure this? And most people ask Colin that and Marianne more than they ask me, but he said, hey, can you measure TMAO? And I was like, what the hell is TMAO? Never even heard of it, right? Okay. So from that time, we've developed some methods to do these analyses, and we've actually done, I wouldn't say 10,000, but probably 5,000 samples. And we do a lot, which evolved just initially from just doing analytical services for other people to actually now designing our own experiments. The first thing that you need to do this is you need a reliable method, just like anything. How are we going to study this if we don't have a reliable method? So for us, it's actually quite easy. We can use a sample as little as 5 or 10 microliters of serum, and people don't tend to believe us when we tell them that. They're like, well, we can send you 100. Is that too much? I'm like, you can send us whatever you want. I'm just saying we only need 5 or 10 microliters, okay? And we use a method called stable isotope dilution, where the analytes that we're looking for, we actually add those analytes into our sample, but they're just labeled with deuterium, okay? And that helps us account and reduce the variability and do quality assurance and things like that. If you want to learn more about stable isotope dilution, you can talk to myself. Colin, I'm sure, knows a ton about that. But actually what we do, and since we've gotten here, we've actually evolved into an even more high throughput platform where it's actually quite simple. We dilute this with acetonitrile, which crashes out the protein. And what we do is load it into a 96 well filter plate and just spin it down into a filter, into a vial plate. Okay, so instead of having individual glass vials that you're filtering and labeling, you just have a 96 well plate. And we use UPLC MSMS. And I wish you could see it here, but we have a method that's about 3 minutes, and in those 3 minutes, okay, we can get our compounds and their D9 labeled forms. And if you're interested in TMA, we can do another step of derivatization, and we can get TMA as well. But so what that gives us the ability to do is we can reasonably do 300 to 400 samples in a day. And my lab people in the back are like, please don't tell people this. Okay. Our record is that we did do over 1,200 in 1 week. Okay. Just because the method is fast and we now have these high throughput vial plates and so forth that we can use. Okay. So because of that, you're going to see the amount of data that we've generated on TMAO from these large clinical experiments that people are like, is it okay if we send you 200 samples? I'm like, yeah, I mean, we can do that in like half a day. It's not a big deal. And they're kind of taken aback by that. But it is quite a simple separation that has allowed us to generate quite a bit of data. So initially we started off working with some people that we had existing samples from clinical studies that we just said, let's just measure TMAO and see if anything pops out of it. So we initially looked at, we said, well, we have this set of subjects where we fed them, we had N of 10 non-obese college students, right? When you work at a place like Virginia Tech, who's the easiest to recruit? College students. They're there. They're poor, okay? They want to be on a clinical study. Perfect, right? I learned this really early at Virginia Tech. If you want to do something, find an area where you can study college students if you're located on a college campus, okay? Anybody ever been to Blacksburg? It's not like Charlotte. There's not a million people here that you can just recruit from. There's like the same 4 people, okay, coming and signing up for your studies, okay? So we had these subjects, and basically what we did is we gave them a lead-in diet, just a control diet, and then what we did is we gave them a high-fat meal, okay? Anybody want to guess what it was? Huh? Close. Well, so it did have a decent amount of protein. We basically got Jimmy Dean breakfast sandwiches, 63% fat, okay? Then what we did is we said, okay, we're going to give them a high-fat diet. So we switched their diet from 55% carbohydrates to 55% fat for just 5 days, okay? It's not a month, not 2 months, okay? Oh, there we go. And then we did that high-fat meal again, and from there we collected samples and we looked at kinetics of trimethylamine production, TMAO production. Okay, so this filled line is their kinetics of TMAO production starting at fasting over 4 hours before they had a 5-day high-fat diet. Okay, we gave them two Jimmy Dean biscuits, and we just looked at TMAO, and you can see it's actually pretty flat, right? It doesn't really change. Okay, but after a 5-day high-fat feeding, okay, not much difference in the baseline fasting, but you can see the excursions of TMAO were very different, okay, and this is, again, just 10 subjects, not a large sample size, okay. So we actually showed here that it doesn't look like a short-term high-fat diet really does much to fasting TMAO, but it actually appears to increase postprandial TMAO. Okay, so this is something that was rapidly modifiable in healthy people. Okay, and I don't show the data. Their weight didn't change. Other things didn't change. That's good news, right? You're like, I can eat whatever I want for 5 days as long as I fast for 12 hours. My weight's not going to change. Okay. Oh, maybe that's just my logic. But we don't really know how this happens. One of the things I will ask you just for thought is, was the substrate even in the colon at 1, 2, 3, and 4 hours? Okay. And I'm going to come back to that question. If somebody, let's limit this to maybe the students. If somebody can come up with a good answer to why you see TMAO excursions, even though it wasn't even in humans, probably in the colon yet, maybe I'll take you out to lunch. How about that? Students are just like, no thanks, I don't want to go to lunch with you. But okay, so this is interesting. This was the first study that we did, and we're like, all right, this is very cool, right? We published this, got a lot of citations. Okay. Again, just a caveat, this is in healthy young men, and I'd like you to take a look at the concentrations that we see at baseline were between 1 and 2 micromolar. Okay? That's very typical for what we see in healthy people. Okay? So the next thing we thought is, okay, well, we've also got this study that was, again, I wish these were clearer, but when you make something on your laptop we had a study where these same investigators said, actually, we had some funding from this Italian probiotic company called VSL, and we did a very similar study, but we supplemented subjects with either VSL3, which is a proprietary probiotic, or control. And so we said, well, if just feeding fat can change TMAO production, imagine what a probiotic can do in terms of preventing that. That's so cool, right? We can actually add live bacteria, and we should get some great changes, okay? So in this case, what we did, again, with the 19 non-obese men, okay, you can get male college students to eat pretty much anything, right? You're going to have a hard, I mean, this may sound sexist or whatever, but men are much more open to eating a hypercaloric, high-fat diet for 4 weeks. Like young engineers are like, I don't care, right? I'll eat whatever I want. So these are the people we could recruit for these studies. But we took them and we did this 12-hour fast, and then we divided them into placebo versus VSL3 probiotic. It's proprietary. I actually don't know what's in it. I just don't know, okay? I would tell you. 9 billion cells, 4 weeks, and in this case, hypercaloric, high-fat diet. Okay, and we did our fasting TMAO again. Now I will say, you just showed me that fasting TMAO doesn't change, so why is this what you did? Well, I didn't know that at the time. I didn't have the data from the first experiment. And again, this was a secondary analysis. I didn't have postprandial samples to look at, okay? These are the initial and final TMAO concentrations for all subjects, and this is the change in TMAO. If you take a subject's if you subtract their initial levels from their final levels, you can see that the high-fat diet increased TMAO production for both arms. Unfortunately, the placebo didn't, or the VSL3 didn't do anything. A probiotic for a month did nothing to prevent these increases. Okay? There were some associations that we saw that changes in TMAO levels were associated with changes in systolic blood pressure and pulse pressure at the carotid site, none in the brachial. Okay, so in other words, the more your TMAO levels went up, the more that your blood pressure and heart rate and things were likely to go up. Okay, but overall, nothing really exciting here. And again, I apologize with the animations. One message I want to give you here is probiotics and prebiotics. Think about this. Probiotics are a very, very, very specific type of bacteria, okay? And a lot of times they don't actually exert global changes on the microbiome, okay? What they do is they affect this very small niche. And just because you affect that very small niche doesn't mean you're going to affect these bacteria that make TMA, okay? So just think about that when people tell you about probiotics or prebiotics can do anything. They're actually affecting a very small niche of the gut microbiome, okay? All right, I want to go through some others because I'll just throw you a spoiler here. The next 5 or 6, or next 3 or 4 studies I'm going to show, we just didn't see any effect of diet, okay? We went back to another, this is actually my postdoctoral boss, Zora Jurich at the University of Michigan. She had this really great Mediterranean diet study. Okay? We know that the Mediterranean diet is associated with reduced cardiovascular disease risk, so we say, well, clearly, we've got to see some TMAO differences here. The Predimed diet, which is a Mediterranean diet study that's still ongoing, showed that a Mediterranean diet did decrease TMAO in Spanish individuals. This was a high HUFA-PUFA diet in Americans. There hasn't been a lot of studies on cardiovascular disease in the Mediterranean diet in American subjects on this scale. Okay, so she actually had 120 people that they recruited and divided them, and instead of just being eat whatever you want versus a Mediterranean diet, they actually did a healthy eating 2010 diet, which is still a healthy diet, versus the Mediterranean diet. So instead of saying Western diet versus Mediterranean, it was actually healthier Western diet versus Mediterranean. Okay, and again, the goal here was to increase polyunsaturated fatty acid intake. And in both groups, increase fruit and vegetable and whole grains. Okay. Well, okay, you can actually see that fish intake went significantly up in the Mediterranean diet group. Both groups actually reduced red meat intake. So you're thinking, oh man, this is awesome. We're going to finally see something. Well, spoiler alert, okay, if you look at TMAO levels here for the healthy eating group, I actually wanted to show all the data points so you can see them for yourselves. okay? Really didn't change anything after 6 months of this dietary intervention. This is not a short intervention, okay? This is 60 people for 6 months in each of these arms, and it just didn't really change anything, okay? One point I do want to make, remember I asked you to remember that those levels in the healthy subjects were like 1, 1.5? These subjects are at higher risk for cardiovascular disease, right? Look at all these values that are up here in the 3s, 4s, 5s, and even greater. So it's not like we're trying to make healthy people healthier. We're taking people that actually could benefit from a reduction in their TMAO levels, but we're just not able to do it, okay? So is fasting TMAO useful for anything? You're like, great, I'm really sick of looking at all these studies where you didn't find anything. If you look, we actually said, well, what if we separate these subjects based on whether they're above or below the median TMAO levels and see how these risk markers correlate with that. And actually, BMI, age, waist circumference, and blood pressure correlated with TMAO levels the way you would expect. So what people would say was, well, your assay, you're just not reliably able to correlate or to measure TMAO levels with a precision and accuracy that you would need to associate it with disease risk. And we showed here, no, that's actually not true. These all hold up with what you would see in the literature. So our lack of effect due to diet is actually real. If there was an effect that mattered, we'd be able to detect it, okay? So therefore, TMAO is useful as a risk biomarker. We just don't know how to reduce its levels except by reducing the substrate intake. A couple of shockers here. Does short-term flavanol supplementation reduce TMAO in subjects at risk for diabetes? No. And again, these are people that have high TMAO levels, okay? How about another one? Okay, let's just try everything we can find. We've tried polyphenols, we've tried fat, we've tried probiotics. Oh, we haven't tried prebiotics. Let's do that, okay? Prebiotics, targeting the gut microbiome with inulin, okay? These are the postprandial curves for subjects before and after inulin intervention. Really no difference. It didn't do anything. And again, remember, prebiotics are targeting a specific niche of the gut microbiome. They don't change the composition and function of the entire microbiome necessarily, okay? So when people are talking about prebiotics and probiotics, remember, these are very specific bacteria. We're not talking about the whole gut microbiome as a whole, okay? So we do have a couple questions remaining in humans, okay? More importantly, utility. We know that TMAO is a good biomarker of risk. We don't really know if that's modifiable by diet, other than saying, hey, let's eat less of the substrate, okay? I'm not going to dwell on this a lot, but one of the things that we think now is that not only do we need to find the correct treatment, we need to find the correct patient. Who is the patient that is able to be benefited from targeting this TMAO pathway? So if you're familiar with the Equal story from 10 or 15 years ago, people that produce Equal versus those that don't from phytoestrogens or urolithins. I know Colin and Marianne do some work with this. There are metabotypes. Some people make them, some people don't, okay? Are we able to target people that are susceptible to responding to our treatment, okay? And I'm actually engaging in emerging collaborations with two of our neighbors next door, Dr. Susan Smith and Dr. Katie Meyer, who actually have existing interests in these areas, so it's been highly synergistic, that we're going to be able to, I think, address some of these questions, okay? I know I'm running short here, so I do want to present something a little bit more interesting, a collaboration that we worked on with Dr. Doug Seals at the University of Colorado Boulder, okay? You want to work with people that have 2 RO1s, like it tends to be fruitful. So when somebody that has 2 RO1s is like, hey, we'd like to send you samples, you're like, yep, send them, I won't charge you anything, right? Okay, so what we did here was a mouse study to look at the effect of aging on vascular health, okay? So what they did here is they have young versus old mice. And they also, within those, either gave them nothing or gave them antibiotic, okay? And they have some really interesting data. So just to show you here, this is the fecal gene copy number. It's not telling you what gene it is, it's just the total number of genes that they were able to measure, okay? And just for reference, Y is young, O is old, and then control versus antibiotic. And you can see in young mice, okay, if you compare young mice to old mice, just aging alone significantly suppressed the number of genes and their copies that were found in the microbiome. Okay? And then within each age group, antibiotics cut it by half or more. Okay? So, and there are some other data here that I'm not going to go into that show that both aging and antibiotics actually change the composition of those, not just their number. Okay? But so again, we know that these antibiotics are significantly altering the amount and the composition of the gut microbiome. And again, okay. One of the things I really want to show you is this curve right here. They added acetylcholine, which basically stimulates dilation of the vessels. Okay? And what they showed was all of these mice had normal response to acetylcholine, except for the aged mice that weren't fed antibiotics. So what they were able to show here is that aging mice don't respond to these endothelial signals to dilate the way that all the other mice do. Okay? Interestingly, you would say, well, what about the aged mice fed the antibiotics? They were just like the young mice. Okay? So this reduced vascular dilation due to aging appears to be microbiome dependent. They were able to show that when you age, you have impaired vascular dilation. When you get rid of the microbiome or significantly blunt it, those deleterious effects are lost. Okay? They looked at pulse wave velocity, which is just arterial stiffness. Okay? And when you look at these young mice, and let's just look at the gray bars, when you compare young to old, you can see the higher your pulse wave velocity, the higher the stiffness of your arteries. Okay? Aging, vascular flexibility was severely hindered. When they gave these aged mice antibiotics, their vascular function, their stiffness improved. Okay? So again, increased arterial stiffness due to aging appears to be microbiome dependent. Same thing you see with inflammation. Older mice had more aortic inflammation. When you added antibiotics, that went away. Okay? They also, and again, I'm not going to dwell on this here, but if you just look at principal cluster analysis, principal composition analysis of the microbiome, you can see that older and younger mice have totally different microbiome profiles. Okay? Okay? And finally, okay, we actually see that if you compare older mice to younger mice, they have significantly greater TMAO levels, and that antibiotics significantly blunted TMAO production. Okay? I really need to work on my animation skills here. Okay? So I just wanted to present that as a little bit more exciting data. This was the first time that it's actually been showed that arterial aging was actually microbiome dependent. And again, this was in animals. They have some studies ongoing looking at humans. But I thought I'd just drop that in here and say not everything that we've done has been not exciting. Okay? That was actually quite exciting. I know I'm running short on time here. I do want to tell you one more thing that we're doing, or not. Okay? So we're looking at ways to reduce TMAO levels. This small molecule inhibitor, DMB, you can see it's actually a structural analog of choline and carnitine, is being explored. There are some patents out there if you go look. We're interested in phytochemicals. I'm not particularly interested. This is a pharma strategy using DMB, right? That's a medicine that you would take. I'm not really interested in that from my research. Some of the things that we're interested in are phytochemicals, okay? And I'm not going to bore you to death here. I wanted to put these figures up here so that you could look. But there's tons of evidence out there. There's all these meta-analyses, right, that show that garlic consumption or garlic supplementation is associated with improved blood lipid profiles, reduced cardiovascular disease risk factors, and reduce lower blood pressure and things like that. Okay, so there's a bunch of meta-analyses I don't want to bore you with here. Honestly, I would bore you with it if I had more time. Let's be honest. I don't really care about your time that much. No, I'm a kid. Okay. But there have been a couple interesting studies in mice where they've shown that if you take mice and you feed them a cholesterol-rich diet, this is basically just cholesterol content of these vascular cells, okay? These open circles are just mice fed this cholesterol-rich diet, and these closed circles are mice fed a cholesterol-rich diet plus garlic extract, okay? Another study in ApoE knockout mice, they're susceptible to these vascular lesions, and you can see adding an aged garlic extract actually reduces the extent of these lesions. Okay? So I think most of you have probably heard, if you've been in the phytochemicals world, you've heard about garlic and cardiovascular disease. Okay? Well, I guess what? This would have been 4 years ago. This paper came out, and what they actually showed was that when you fed mice, so they fed mice for 6 weeks with either chow plus carnitine, and then chow plus carnitine plus allicin, okay? And they actually showed, if you look at the blue line is the chow diet. This is a carnitine challenge test where, similar to what we're doing with the meal challenge, they administered carnitine and looked at blood levels of TMAO out to 8 hours. You can see that the chow diet, or the control, is the blue line. When they added carnitine, you can see that TMAO production just goes through the roof. And then adding allicin to the carnitine dropped it back down to the yellow level. So allicin, this garlic sulfur compound, was significantly able to inhibit TMAO production. Now, there's really nothing about mechanism here other than the fact that they did show that TMAO production was associated with certain groups of bacteria. Okay, so one of the things that we're working on then, and again, I keep forgetting to do this, and I really apologize. So one of the things that we're interested in is what is the mechanism by which this happens, and what are really the active compounds that that are responsible for this effect. Okay, so in garlic, if you just look at garlic itself, when you pick it, when it's sitting in the store, the intact cloves have this compound called, I call it alliin. Does anybody call it something else? Alline? I call it alliin. Right? When you decompartmentalize it, when you chop it up to make that nice shrimp scampi or whatever it is that you're making at home, this enzyme, just like polyphenol oxidase in apples, becomes decompartmentalized, and you have access to oxygen, and you get conversion of alliin to allicin. And almost all the studies that have been done on these aged garlic products. In other words, chopped garlic, where you have primarily allicin and less alliin. So what we want to know is which of these compounds is actually the active compound, and why does this matter? Well, okay, if in an industrial or a food setting, if you're crushing up or decompartmentalizing your garlic before you're cooking with it, or in this case freeze drying it to make a garlic powder, then you have an allicin-rich product. Okay? But you could, in theory, inactivate that enzyme either by steaming, or you could just freeze dry the cloves whole. Okay? And you could get an alliin-rich product. So the question is, from a foods perspective and supplementation perspective, which one of those is better? Okay? We don't know. So we have an experiment that my graduate student Mike, who is a Virginia Tech student, and he's actually wasn't able to be here today because he has to go to class and stuff like that, whatever. But he's doing, this is his PhD work. We're looking at whether allicin or alliin is more effective and the mechanisms by which this happens. Okay? So what he did with the help of my students that are here today, we actually made garlic extracts, and these are the LCMS traces. The scales are normalized to the same level, so you can see relative abundance. This is the fresh garlic extract, and you can see that there's actually a lot more alliin than allicin. When you have the aged garlic extract, you can see there's still an appreciable amount of alliin there, but your amount of allicin has gone through the roof now. Okay. And then we're doing this, and I do need to wrap up here, but we're doing this in a model. And one of the things that I learned, I went to nutrition this summer, and I was sitting in a conference and in a symposium, and a speaker got up and absolutely unleashed hell on the field. Okay, right? And and if you go back and look, this is the guy that has studied. He's probably the world's expert on flavin monooxygenase 3. And he just made a statement that said, oh yeah, you guys need to use female mice instead of rats or male anything. And we're just like, what? Okay, right? So, and what I didn't know about this was this FMO3 varies quite a bit. And FMO3 is this enzyme that basically reacts with tertiary amines and reacts with thiols. Okay, and it's again the same enzyme that if you lack it, you have this fish odor syndrome. Okay. And he presented some data, and we went back and we couldn't believe it, and we looked it up. And actually, if you look at FMO3 protein and mRNA expression between male and female mice, do you see anything there for FMO3 for the males? Very low, okay? And actually, that's kind of some similar data. If you look at rats, okay, there's less of a difference between sexes, but actually the absolute levels are much higher in mice than they are in rats. Okay? So all of us were sitting there planning on doing these experiments in male mice like we normally do, or rats even, which is myself and Michael Keenan from LSU had studies planned using male rats, and we had to come home and completely redesign the experiments based on what this talk had said. Okay? So we are in fact using female C57 black 6 mice, and we're going to do some experiments where we're going to do a carnitine challenge to look at TMAO kinetics before and after the experiment. We're also going to look at levels of TMA-releasing bacteria, the actual CNTAB enzyme that releases TMA. We're going to do shotgun metagenomics to look at overall community function. And then what we did is we actually divided these into a variety of treatments, basically seeing if allicin-rich or alliin-rich garlic extracts can reduce the effects of carnitine at increasing TMAO production. And then at the end, we're going to do the carnitine challenge and the fecal samples again, and also look at liver FMO3. Okay? So actually, this study, the animal portion of it has been completed. We're just doing sample analysis. I really wish I had some data to share with you, but I did want to let you know where we were going with that. So basically, our future directions are based on the results of this study and some other things. If we do see that garlic is associated, that one compound is more associated with reduction in TMAO levels, is that then associated with reduction in atherosclerosis in an animal model? Okay, and by what mechanism? The other thing we're doing, relating more to humans, is this idea of individualized medicine, which kind of dovetails really nicely with what the UNC folks over at NRI are doing, is trying to figure out who are the people that we are able to treat or reduce TMAO production and reduce disease risk with some of these dietary interventions. Are there people that no matter what we do, we're not going to be able to reduce their TMAO levels? And are there people that are likely to be benefited by some of these interventions? Okay? So we can use some different models to identify perhaps polymorphisms in FMO3 gene. Are there certain gut microbiome profiles that lend themselves to inhibition of TMA release? Is there FMO3 status that we can identify in vivo that says, yes, this person might actually be susceptible to, or not susceptible, but benefit from some of these treatments, so that we can actually target some of these dietary interventions to people that will actually benefit from it, right? Wouldn't that be useful if somebody could say, hey, actually, I know for a fact because of these different traits that you have, that this treatment is actually going to work better for you, or this dietary intervention is going to work better than this one, so don't waste your time with it. Okay? Okay, I am still technically within the hour time, so I don't feel too bad. I did want to make sure to do an acknowledgement and acknowledge all the people that have really, really helped with this. Students and faculty at Virginia Tech. David Rains is if you look at, so 3 of these people, actually 4 here, Laura Griffin, Kat Racine, Mike Hughes, and Lauren Essemacher. While they've been at Virginia Tech, they've also been here. David is the only one, he's by himself here because he's the only one that hasn't been at Virginia Tech as well. And then folks from University of Colorado Boulder, USDA, and the University of Michigan. And these are just some photos. I couldn't find a photo that had David, he's too new here. But again, you know, these are the folks that really make it happen. I sit up in the office and complain about data and things like this, but these are the folks that are actually doing things, generating data, and to quote our little strange planet, actually doing science, right? I'm just talking about science. They're the ones actually doing it. Also to acknowledge our funding. I haven't been in the PI on any of these TMAO grants. Hopefully in the future that will change. Great news, actually, we did just get a 9th percentile score on R21, so hopefully next year we'll have some more cash from NIH. With that, I appreciate you guys listening to me ramble on here about TMAO and the gut microbiome, and I'd be happy to take any questions that you may or may not actually have.